Welcome back, everyone, to this part of the topic on the different types of blood vessels. And in terms of learning objectives, they're to be able to describe a capillary bed and explain how it regulates blood flow, describe the structure and function of the three different types of capillaries, and finally, be able to explain the features of veins, including the structure and function of venous valves in the large veins of the extremities. So a quick recap on what we just learned. We learned about elastic arteries. Remember, they're the ones that are closest, to the heart, and they're under high pressure with a maximum of 120 millimeters of mercury. And they contain those elastic fibers, which allows them to expand and recoil. And that expansion then is called that compliance, okay? So the aorta is a compliant vessel. Muscular arteries then have less elastic tissues, but lots of smooth muscle. These are called distributing arteries because they regulate the flow of blood to particular body regions. And then those smaller muscular arteries and the arterioles then are small arteries that are called resistance arteries because they regulate the flow of blood into capillary beds through the property of vasoconstriction and vasodilation. Remember, vasoconstriction will increase peripheral resistance and therefore blood pressure. So we're going to talk about these capillary networks now, but I want you to have a look at this figure over here on the top right. You can see here the aorta coming out there, the left side of the heart, and then it's responsible then pretty much, for delivering that oxygenated blood into the systemic circulation. You can see all of these branches then of the aorta delivering blood to different body regions. So here we've got the upper body. Of course, here we've got the delivery to the brain. We've got also delivery to the liver here, to the kidneys, and then to the lower body here. So what you have to imagine, right, is from this aorta, it branches into other arteries, which branch, eventually into these arterioles, and then those arterioles feeding into the capillary bed. Now, the capillary beds contain capillaries, and remember that it's across the capillary walls that you have the exchange of gases and wastes and nutrients. Now, the capillary beds and these capillaries contain pores to allow this to occur. And there are different capillary beds in different parts of the body. For the most part, the capillary beds just allow the exchange of gases and ions and some nutrients. But in some parts of the body, these capillary beds have really, really large pores, right? So in places where, you need the passage of proteins or cells. So say, for example, in the bone marrow or in the liver, you would have these sort of really leaky capillaries. And in a couple of slides' time, we're going to go through the different types of capillaries that can be found in these capillary beds. All right, let's go back here to this figure here. So you can see here in a capillary network, as I mentioned right at the last part of the previous recording, we have these precapillary sphincters, which are smooth muscle, and they're found in those metarterioles, and they therefore can regulate blood flow through the capillary beds. Right, so this is smooth muscle. When it contracts, it's going to cause vasoconstriction, and therefore there's going to be decreased flow of blood through this capillary bed, right? If the precapillary sphincter smooth muscle cells relax, that's going to open up the capillary bed, and you're going to get increased flow due to that vasodilation there. Now, the flow through the thoroughfare channel, which you can see here, is fairly consistent, while the flow through those arterial capillaries then is intermittent. So just to repeat that, when the precapillary sphincter is contracted, this capillary bed is kind of shut off. So this metarteriole then can serve as this vascular shunt to shunt blood there into the venous system. And that's because these precapillary sphincters are closed. But then when those smooth muscle cells there relax to cause, vasodilation, that's going to literally open up those capillaries and blood will flow through them. So we're going to now have a look at what conditions might promote the opening up of the capillary bed, and another condition which will, keep it closed off. So I'm sure some of you have already thought of this example. It's what happens on a hot day, right? So we're talking about the microcirculation here, and the microcirculation involves the arterioles, the capillary bed, and these small vessels here on the venous side called the venules, which I'll get to in a few slides' time. So on a hot day, and in particularly if you're exercising on a hot day, our core temperature starts to increase. Now the body needs to get rid of that excess heat to maintain homeostasis. So what happens is that blood flowing through the arteriole, then into the meta-arteriole, we have the precapillary sphincter relaxes, and that literally opens up the capillary bed so that blood can flow into the capillary bed or perfuse the capillary bed. And these capillary beds are near the skin surface, right? So that's then going to allow that heat then to just dissipate into the environment, and our core temperature, goes back to normal, and homeostasis then is maintained. However, on a cold day then, you can see very clearly that the capillary beds in the skin, they're not open at all, right? And you can see that the blood flow here is kind of shunted through these meta-arterioles. You can see that thoroughfare channel there forming and connecting that, that on the arterial side then to the venule on the venous side. And that's because blood doesn't need to be cooled as much near the skin surface. And finally, another really good example of how the capillary beds regulate flow according to demand is during exercise. You can see all the percentage of cardiac output here that's delivered to all of these organ systems at rest, and then during vigorous exercise here. And you can see a very good example here that during exercise, blood flow to the gastrointestinal tract is literally shut down, and you can only see 1% there of cardiac output going there. And that is due to the vasoconstriction of those vessels preventing flow to the GI tract. On the other hand, though, all of that, extra blood flow is going to as exercising muscles, and that's because of vasodilation of those vessels to the muscles. And it kind of all makes sense, doesn't it? So we're now going to talk about the 3 different types of capillaries, but just talking about capillaries in general to begin with. They are, of course, the smallest blood vessels. Their walls consist of a thin tunica interna or intima, and believe it or not, they are just 1 cell thick. Amazing, right, when you consider what's going on across the walls of these little capillaries. And they carry blood from arterioles to venules. And as I mentioned on the previous slide, together the arterioles, capillaries, and venules constitute the microcirculation. Now, capillaries are not evenly distributed in the body, and the highest numbers are found in tissues with a high metabolic rate, so say the kidneys or the liver or skeletal muscle, to name a few. And they are absent, of course, in avascular tissues such as cartilage, and the word avascular just means without blood supply, right? Now, substances move through capillary walls via diffusion, straight through the plasma membrane for lipid-soluble and water-soluble molecules, so say lipid-soluble vitamin A, D, E, and K, and then through these fenestra, and the word fenestra is Latin for a window. So these are these larger sort of gaps between endothelial cells in some capillaries. And this will facilitate then the movement of larger sort of water-soluble molecules such as proteins. So if you have a look at this figure up here, and it doesn't matter what the type of capillary is at this stage, but you can see that the endothelial layer is just 1 cell thick. So that's the tunica intima there. And then we've also got a basement membrane as well. But if we then look at the 3 types of capillaries, we have continuous capillaries, we have fenestrated capillaries, and you can see the difference there, all of those gaps appearing between, the endothelial cells, and you can see they're sort of studded along the length of that capillary. And then we also have these sinusoidal ones where there is massive holes. You can see an incomplete basement membrane there, and you can see much bigger gaps there between the endothelial cells. So we're going to talk about all of these three types now. The continuous capillaries look like this. There's no gaps between the endothelial cells at all. So there's no fenestra, right? They're less permeable to large molecules compared to other capillary types. And where do we find these ones? Well, in places like muscle and the skin and nervous tissue. And these continuous capillaries of the brain have tight junctions completely around the endothelium, and these capillaries constitute the blood-brain barrier, right? So what I want you to get from this slide is with, the continuous capillaries, they're less permeable to large molecules. So you're going to get passage of way less stuff through the walls of these continuous capillaries. And so we're going to call them not leaky capillaries, right? If we move on now to the fenestrated capillaries, and you can see them all, the fenestrations there. So these have many pores between the endothelial cells, or gaps, called fenestra. These are highly permeable, right? And they're found wherever active capillary absorption or filtrate formation occurs. So what does that mean? So capillary absorption occurs in the intestinal villi, right? We'll get to that more when we get to digestion, but think about the end products of digestion need to be absorbed into capillaries. And that's why we have these fenestrated capillaries in the intestinal villi. The glomeruli of the kidneys. Now, where we have the glomerulus is where the blood is filtered. And of course, that's going to require as well these fenestrations as well, because you're going to get things like glucose and other sort of nutrients getting filtered into the filtrate across the wall of the glomerulus, which is a network of capillaries, and they are obviously fenestrated capillaries, right? Then we have the endocrine glands as well. And we talked about that. We know about hormones. And these fenestrated capillaries are characterized by greater permeability than other capillaries. So we're going to call these ones leaky, okay? Now we get to the sinusoidal capillaries, right? These are highly modified, okay? They have a big, large diameter. They're very leaky. They have fenestrated, they are fenestrated capillaries with really large lumens. And where do we find these? We find them in places like the liver, the bone marrow, and endocrine glands as well. And they allow the passage of large molecules, so proteins and even blood cells, like if you think about the bone marrow, to pass between the blood and surrounding tissues. Now, blood flows sluggishly through these sinusoidal capillaries, allowing for, modification then in various different ways. Okay, so we are going to call these ones super duper leaky, okay? So they are the three types of capillaries: the continuous capillaries, the fenestrated capillaries, and the sinusoidal capillaries, or the not leaky, the leaky, and the super leaky. We're going to finish up now talking about the veins and the venous system in general. Remember that there is lower pressure in the venous system compared to the arterial system. And the pressure is about 10 millimeters of mercury, and it doesn't fluctuate very much. Compared to arteries, we've mentioned this before, veins have thinner walls and they have less muscular and elastic tissue. Now, because the walls are thin, it means they can expand easily, okay? So we say that veins have high capacitance, or they are high capacitance vessels. Now, the word capacitance, which is also termed compliance in some textbooks, but capacitance means that they can expand readily to hold a high volume of blood, even when, pressure is low. And if you think about all of the, the blood in the body, and I'll show you this on the next slide, there's about 84% of total blood is in the systemic circulation, and 64% of that is actually in the systemic veins. Now, we also have veins that have valves, which aid skeletal muscles then in making sure that blood flows in an upward direction. So particularly venous valves in those veins of the lower limb, which ensure one-way flow of blood back towards the heart. Okay, we have these venules. We've talked about these a little bit in terms of the capillary network. These are these little vessels that drain the capillary networks. They contain endothelial cells and basement membrane with a few smooth muscle cells. And as the diameter of the venules increases, then the amount of smooth muscle increases. So what you have to imagine is these venules drain the capillary beds, and then they start to emerge. And then as they're merging, they become larger venules, and eventually they become these small veins. And with the small veins then, there's smooth muscle cells form that continuous circular layer. There's an addition of a tunica adventitia or externa, which is made of collagenous connective tissue. If we look at how veins can act as blood reservoirs, here in this table you can see that the percentage of blood that's in the systemic circulation. So predominantly in the systemic circulation, 84%, 9% in the pulmonary circulation, and 7% in the heart. Of that 84% in the systemic circulation, 64% of it is in your systemic veins, 13% in the systemic arteries, and 7% then in the systemic capillaries. Of that 64%, then 18% in the large veins. We have these venous networks that are present in the liver, the bone marrow, and the skin, and they can hold 21% of that blood, and the venules and the medium-sized veins 25%. And you can follow this, and look at it in your own time. Now, if blood needs to be redistributed to different sort of parts of the body, and the vasomotor center in the medulla oblongata in the brain causes sympathetic stimulation of smooth muscle in the walls of these veins. And this is called venoconstriction, right? When the smooth muscle in the walls of the veins contracts, it causes venoconstriction. What this does, it increases pressure on the blood within the veins, and that will accelerate or speed up the return of that blood then to the heart. Now, if you remember in this table, I mentioned that about 21% of venous blood is located in these venous networks, right? And this is termed the venous reserve. And through the process then of venoconstriction, this reserve volume of blood can return to the heart way more quickly and then be redistributed to other parts of the circulation. So I guess what I'm trying to say here is this blood here in the networks can kind of be called upon if it needs to be redistributed to other parts of the circulation. So say when you're exercising. Final thing now we're going to look at are the presence of these valves in veins greater than that have a diameter greater than 2 millimeters. So because the venous system receives less pulse pressure and often works against gravity, additional structures and pumps are required to promote venous return to the heart. These little valves are found in veins, as I mentioned, with diameters greater than 2 millimeters, and they stop the backflow of blood. So these venous valves are far more common in veins of the lower limb because, because of gravity, you're trying to push blood in an upwards direction back towards the right side of the heart, but gravity is going to tend to cause backflow. And in that case, the little valves sort of help you to prevent that by closing over. As you can see here, we've got action of the skeletal muscle pump as well. So that contraction of skeletal muscle there will help to move blood. So here we've got our muscles relaxed and the valves are closed. And here we've got when the skeletal muscle contracts, that's going to push that blood open the valve and the blood will be pushed in an upwards direction. And finally then we have the respiratory pump and movement of that sheet of muscle, skeletal muscle being the diaphragm, will help to pump blood then through the abdomen. And I'm sure that some of you have heard of a clinical condition known as varicose veins. And varicose veins occur because of these defective valves that don't close over properly in the lower limb, and that causes backflow of blood, and that backflow of blood then is going to increase hydrostatic pressure in the lower limb, and that leads to sort of the twisting of these superficial veins to cause painful varicose veins. And I'm sure as you move through your course, some of you will learn a lot more about how they develop. Okay, everybody, I have actually finished. I'm going to leave you with this table here, which is a summary of arteries and veins. You can follow it yourselves in terms of pressure differences, the valves, only being present in the veins, not in the arteries, the direction of blood flow, the general appearance, and the thickness of the walls. Okay, and as usual, we have a couple of summary slides to help you with your study. So it's